All right, this section deals with the genetic code and protein synthesis. By 1961, it was clear that the sequence of bases in DNA served to direct the synthesis of mRNA, and that the sequence of bases in mRNA corresponded to the order of amino acids in a particular protein. However, the genetic code, the exact relationship between mRNA sequences and amino acids, was unknown. At least 20 mRNA words are needed to represent the uniquely 20 common amino acids that we find in proteins. If the mRNA words consisted of a single letter represented by a base, A, C, G, or U, we could only have 4 amino acids that could be uniquely represented. Thus, it was proposed that it is not one mRNA base, but a combination of bases that codes for each amino acid. For example, if a code word consists of a sequence of two mRNA bases, there are 4 to the power of 2, or 16 possible combinations, so only 16 amino acids could be represented uniquely. This is a more extensive code, but it still contains too few words to do the job for 20 amino acids. If the code consists of a sequence of 3 bases, there are 4 to the power of 3, 4 times 4, gives you 16 times 4 gives you 64 possible combinations, more than enough to specify uniquely each amino acid in the primary sequence of a protein. Research has confirmed that nature does indeed use three-letter code words, a triplet code, to store and express genetic information. Each sequence of 3 nucleotide bases that represents code words on mRNA molecules is called a codon. And so you can see this sequence of amino acids here. And then we have a codon for the methionine, which is AUG. Then we have one for the valine, then one for the lysine, then one for the valine. But you'll notice that these two codons here for valine, you'll notice they're not identical to each other. In 1961, scientists at the National Institutes of Health attempted to determine which three-letter codons corresponded to each amino acid. They made a synthetic molecule of mRNA consisting of uracil bases only. Thus, this mRNA contained only one codon, the triplet U, U, U. They incubated the synthetic mRNA with ribosomes, amino acids, and tRNAs, and the appropriate enzymes for protein synthesis. They obtained a polypeptide that consisted only of phenylalanine. Thus, the first word of the genetic code had been deciphered. U, 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 or again, uracil, 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 coded for phenylalanine. And you can see that one right here. We go from U to U to U, and we end up with phenylalanine. A series of similar experiments by other researchers followed, and by 1967, the entire genetic code had been broken. The complete code is shown right here. Now, going back to the previous slide, the genetic code has several important characteristics. First, it applies almost universally. With very minor exceptions, the same amino acid is represented by the same three base codon or codons in every organism. Second, most of the amino acids are represented by more than one codon and are said to be degenerate. And so valine, as you can see here, would be degenerate because we have two different sequences of the three GUC and GUU that code for valine. Only methionine and tryptophan are represented by single codons, as you can see here. Leucine, serine, and arginine are the most degenerate, with each one being represented by a possibility of 6 different codons. The remaining 15 amino acids are each coded by at least 2 codons. Even though most amino acids are represented by more than one codon, the reverse isn't true. No single codon represents more than one amino acid. Each three-base codon represents one and only one amino acid. Only 61 of the possible 64 base triplets represent amino acids. The remaining three, UAA, UAG, and UGA, are signals for the chain terminations and are called stop codons. You see, I have them circled here. They tell the ribosome when the primary structure of the synthesized protein is complete and it's time to stop adding amino acids to the chain. Now, the presence of stop codons in the code implies that start codons must also exist. There is only one initiation or start codon, which I have circled in red, AUG, which is also the codon for the amino acid methionine. AUG functions as an initiation codon only when it occurs as the first codon of a sequence. So up to this point, you've become familiar with the molecules that participate in protein synthesis and the genetic code, which is the language that directs the synthesis. But now we're going to take a look at the actual process by which the polypeptide chains are put together. There's three major stages in protein synthesis, and we're going to start with the initiation of the polypeptide chain. Then we're going to follow that by elongation of the chain, and then finally the termination of the completed polypeptide chain. The first amino acid that starts the process of protein synthesis in prokaryotic or bacterial cells is a derivative of methionine. This compound, called N-formyl methionine, initiates the growing polypeptide chain as the N-terminal amino acid. Most proteins do not have N-formyl methionine as their N-terminal amino acid, however, which indicates that N-formyl methionine is cleaved from the finished protein when the protein synthesis is completed. A ribosome is a large complex consisting of both protein and RNA. It has two subunits, a large subunit and a small subunit. The initiation process begins when mRNA is aligned on the surface of a small ribosomal subunit in such a way that the initiating codon, AUG, occupies a specific site on the ribosome called the P site or the peptidyl site. When the AUG codon is used this way to initiate synthesis of a polypeptide chain in prokaryotic cells, it represents N-formyl methionine instead of methionine. When AUG is located anywhere else on the mRNA, it simply represents methionine. For the eukaryotic cells of humans, AUG always specifies methionine, even when it's the initiating codon. Next, a tRNA molecule with its attached N-formyl methionine binds to the codon through hydrogen bonds. The resulting complex binds to the large ribosomal subunit to form a unit called an initiation complex. In the next phase of elongation, the whole ribosome moves one codon along the mRNA toward the 3' end. As the ribosome moves, the empty tRNA is released from the P site, and tRNA attached to the peptide chain moves from the A site to the P site. This movement of the ribosome along the mRNA is called translocation and makes the A site available to receive the next tRNA with the proper anticodon. The amino acid carried by this tRNA bonds to the peptide chain, and the elongation process is repeated. This occurs over and over until the entire polypeptide chain is synthesized. The chain elongation process continues and polypeptide synthesis continues until the ribosome complex reaches a stop codon, UAA, UAG, or UGA, on the mRNA. At that point, a specific protein known as a termination factor binds to the stop codon and catalyzes the hydrolysis of the complemented polypeptide chain from the final tRNA. The empty ribosome dissociates and can then bind to another strand of mRNA to once again begin the process of protein synthesis. Several ribosomes can move along a single strand of mRNA one after another. Thus, several identical polypeptide chains can be synthesized almost simultaneously from a single mRNA molecule. This markedly increases the efficiency of utilization of the mRNA. Such complexes of several ribosomes and mRNA are called polyribosomes or polysomes. Growing polypeptide chains extend from the ribosomes into the cellular cytoplasm and spontaneously fold to assume characteristic three-dimensional secondary and tertiary configurations. All right, example 13.5, write the primary structure of the polypeptide produced during translocation of the following mRNA sequence. So in order to answer this, we have to go back to the genetic code. All right, so we're going to start with the AUG. So AUG, so this is methionine. So we're going to have our 5', then we're going to have our methionine. Then we have UUG. So let me just erase this here. So we have U, U, and G. So that would be a leucine. So we'll add the leucine like that. After that one, we have GAG. So we have G, A, and G. And so that's a glutamic acid. So we use GLU to represent glutamic acid. All right, I'll erase all that. Next, we have AAA. So for AAA, we have A, and then A, and then A. And so that's lysine. So we'll put lysine here. All right, erase that. Next, we have GUA. So we have G, U, and A, and that's a valine. So we'll put our valine. And then finally, we have GGA. So for that, we have G and G and A. That's a glycine. So we'll scribble that down, GLY. And there's our 3' end. The base pairing mechanism that we saw in chapter 13.4 provides a nearly perfect way to copy a DNA molecule during replication. However, not even the copying mechanism involved in DNA replication is totally error-free. It's estimated that on average, 1 in every 10 to the power of 10, which is a big number of bases of DNA, so 1 in 10 billion, is replicated incorrectly. And any change resulting in an incorrect sequence of bases on DNA is called a mutation. The faithful transcription of mutated DNA leads to an incorrect base sequence in mRNA. This can lead to an incorrect amino acid sequence for a protein, or possibly the failure of a protein to be synthesized at all. The three main types of mutations that result in translatable damage to DNA are called substitution, insertion, and deletion. In base substitution, a single base is substituted for another base. For example, if a CTT is mutated to a TTT in the coding region of DNA, then the amino acid in the resulting protein changes from a glutamic acid to a lysine. In a base insertion mutation, extra nucleotides are added to a DNA region. This changes the reading frame such that the protein product made is truncated or non-functional. In a deletion mutation, one or more nucleotides are deleted from DNA. This also changes the reading frame, which often leads to a non-functional protein product. And if we take a look at our normal sequence of amino acids here we have a substitution where we change this GAA to an AAA. So we change the GLU to a lysine. So we change the glutamic acid to a lysine. Then you see we have an insertion here. Okay, so we're inserting the wrong one here. And then we have a deletion, whereas if we're removing something. Some mutations occur naturally during DNA replication. Others can be induced by environmental factors such as ionizing radiation like X-rays, ultraviolet light, and gamma rays. Repeated exposure to X-rays greatly increases the rate of mutation. Thus, patients are given X-rays only when necessary, and technicians who administer X-rays remain behind protective barriers. A large number of chemicals like nitrous acid and dimethyl sulfate can also induce mutations by reacting with DNA. Such chemicals are referred to as mutagens. Some mutations might benefit a species over time as mutated DNA gives rise to varying characteristics, causing individuals to survive better in their environment. Mutations may also be silent in that they do not affect the translated protein and therefore have no effect on an individual organism. Conversely, mutations may be lethal or produce genetic diseases whenever an important protein or enzyme is incorrectly synthesized. Sickle cell disease, phenylketonuria, hemophilia, muscular dystrophy, and many other conditions are the result of such mutations that have become permanently incorporated into the genetic makeup of certain individuals. In sickle cell disease, a DNA base mutation causes a hydrophobic valine to replace a hydrophilic glutamic acid in hemoglobin. This single mutation makes the protein fold differently. The adjacent hemoglobin peptide chains try to pack together to prevent exposing the hydrophobic valine to the aqueous environment, which is our blood. And as the hemoglobin chains stack together, they form long rod-like structures inside the red blood cells that warp the shape to resemble that of a sickle. The sickled blood cells have a tendency to clog blood capillaries, which has many catastrophic side effects such as anemia, vision problems, and even strokes.